Good evening, my name is Hunter, and this is content review for Principles of Biology 211 students. This is week 9, DNA replication, transcription, and translation. I'm delivering this at an unusual time because it is the week of a holiday, and that will interfere with our usually scheduled broadcast. This video can be found uploaded on the PSU Learning Center's YouTube channel at any time. I am not sure how this is going to affect deadlines for extra credit. I will endeavor to make sure that everyone gets the credit that they should get. So with that, this is the central dogma of modern biology. This is so important. I really like this section. I have so many flashcards from my time in Principles of Biology learning this section. Um, it is that important. Um, so let's get into the material. An E. coli cell that contains a single circular chromosome made of double-stranded DNA is allowed to replicate for many generations in nitrogen-15. This is a heavier isotope than is usually found. That is the important thing you need. That is an important detail that you need to remember, in fact. Nitrogen-15 and 14. It's allowed to replicate for many generations in nitrogen-15 medium until all of the E. coli cell's DNA is labeled with nitrogen-15. One E. coli cell is removed from the nitrogen-15 medium and is placed into a medium in which all of the available nucleotides are nitrogen-14. Nitrogen-14 labeled, yes. The E. coli cell is allowed to replicate until 8 E. coli are formed. Given the situation, which of the following is true? A, some of the nitrogen-15 labeled DNA will be found in all 8 cells. B, some nitrogen-15 labeled DNA will be found in only 4 of the cells. C, some nitrogen-15 labeled DNA will be found in only 2 of the cells. Or D, some nitrogen-15 labeled DNA will be found in only 1 of the cells. So it's basically if we distill this question down, there's a lot of details here, but if we get to the core of it, is parent DNA marked with nitrogen-15 going to appear in all of the cells, half of them, only 2 of them, or only 1 of them? So let's look at, we have generation 1. I'm going to draw these as linear strands of DNA, 2 linear strands of DNA, even though this would be a circular double-stranded DNA. So that is our parent strand of DNA. And to answer this question, you need to know which model of DNA conservation we're talking about. And by conserving, I mean how much of parent DNA and what parts are conserved from generation to generation as it's replicating. So let me get another color going here. Basically, I'm going to split these down the middle is what's going to happen. And this is called, my undo button is not working well. Okay. So if it was fully conserved, both strands would stay together, and then the daughter strands would pair together and form a completely new double helix. That is not what happens. In the semi-conservative model, which we know to be correct because of this nitrogen pairing test, this nitrogen isotope test, the daughter DNA has 1 strand of parent DNA paired with a strand of replicated daughter DNA. I think I'm using the right vocabulary here. So each of those parent strands is replicated and paired with new DNA. So this represents 2 new double-stranded circular chromosomes. Okay. If we get another replication, we split each of these down the middle again, and they replicate. Each of those, I kind of want to do these in a different color to show the next generation. We don't really need to. Oh, back up. Come on. So here we have 4 chromosomes. This is an additional replication. And then for our final one, each of those splits again. One, two, three, four, 5, 6. I'm going to run out of room. 8. 8 chromosomes. The solid lines denote our nitrogen 15 labeled chromosome or genetic material. And the dotted lines are our nitrogen 14 marked materials, genetic material. This is the experiment that proved that the semi-conservative model of DNA replication, but this experiment served as the best evidence we have that the semi-conservative model of DNA replication is how DNA replicates. So this is a good experiment to know. It's good to understand. So based on this, C is our final answer. Some nitrogen 15 labeled DNA will be found in only 2 of the cells. And I'm going to draw this in another color or highlight this. Those 2 chromosomes or those 2 cells. God, my tablet is behaving just slow enough that it's going to make me a little crazy. Come on now. There. Okay. Draw a model of a short segment of double-stranded DNA. Your model should describe the roles and relationships of all of the following enzymes and structures in replication. All right, this is a big ask. There's a lot going on. So let's start out. I'm going to have this with my, there we go. Okay, I have this list pulled up on another screen so that we can zoom in here and really, I hope, get into the details here. I'm using, there we go. Okay, we'll start in black. Come on now. All right, let's start with our parental DNA. We're going to have a strand that starts here, and this branch will make sense in a second. And then we're going to have its corresponding partner in the double helix. 5' defines one end. 3' would be the other end. Now DNA is anti-parallel, and that means that they're facing opposite ends, basically. Where one starts, the other begins. And then we have our base pairing between them. And I've put my 5' there, so I need to think about the direction of replication here for a second. Overall direction of replication would be in both directions, but this is our leading strand. So I want to do this in a different color. So red is going to be our new DNA going in this direction. Yes. So we're always going in the 3' to 5' direction is the overall direction of replication. We also have these shorter strands called Okazaki fragments. Yes. This is our leading strand. These are our lagging strands. We've already got our anti-parallel strands. We have our single-stranded DNA. Let's do, these are our Okazaki fragments. I want to see how they labeled this. Eh. Okazaki fragments. So next, let's work on some proteins. We'll do these in blue, please. Thank you. All right. So here we have DNA polymerase. DNA polymerase can only go in one direction, can only go in that 3' to 5' direction. So to go back and do these Okazaki fragments and tie them together, a DNA ligase has to hop back. So where do I want to put that? Yeah, we're going to join these two fragments. Is joining those broken fragments of replicated DNA together. Let's see. We're going to make some room here. This is going to be DNA primase. Primase is getting the parent strand ready for replication to follow. So it's laying down building blocks, for basically laying down the tracks that it needs. We'll call this one helicase. Helicase is unwinding the helix. It's relieving some pressure, some of that tension. Let's see. Yeah. You know what? I'm going to change this one because this is a better visualization for one of the other polymerases. Let's see. No, I'm going to leave it as ligase, joining those fragments together. What else do we got? Helicase, DNA polymerase 3. DNA ligase. Primase lays down primers. We did binding proteins. We did 3' to 5' direction. I'm missing something important here. Come on, there we go. Origin of replication is where that DNA polymerase attaches and starts laying down DNA. Let's see. This whole thing, generally speaking, would be a replication bubble. What else is important to show? Replication fork basically refers to the fork, the branching in the DNA that forms. Here I'm just drawing my base pairs. I see RNA primers on the list, but I can't think of a good way to show that right now. Hmm. Okay. Yeah, I think I'll leave it at that for now. Moving on. Some researchers estimate that the mutation rate for a given gene, or its DNA, in certain strains of bacteria is about 10 to the negative 8. This means that one error or mutation in a given gene is introduced for every 100 million cell divisions. What can cause mistakes in replication? Yeah, this is a good opportunity to type. All right. So what can cause mistakes in replication? An incorrect or mismatched nucleotide. There. Can be added during replication. You could have X-rays, UV light, or certain chemicals can modify the DNA so that errors are perpetuated into subsequent replication events. Yeah, I think that about covers it. How are such mistakes normally corrected? So DNA polymerase. DNA polymerase is really important here. Polymerase inserts new nucleotides during normal replication. It also checks or proofreads each new nucleotide against the template for mismatches. Normally. Come on now. each new nucleotide. For mismatch. And if a mismatch is found, DNA polymerase can then remove it and replace it with the correct nucleotide. Errors introduced by X-rays and other factors are generally repaired by other DNA repair enzymes in the cell. Um, okay. Next up, draw a model for intron removal, which contains pre-mRNA, exons, RNA splicing, spliceosome, introns. So introns and extrons are ways of referring to portions of DNA that get expressed or not. And the way that I remember this is extrons, or exons rather. Introns and extrons. Yeah. Intron, exon. Exons get expressed. Oh, come on. Oh no. Stand by. I've made a terrible mistake. Oh no. Come back. Okay. Are we back? Dang it. I hit the wrong button and now I need to refresh my screen connection. 1 second. I have been muted this entire time. That is really embarrassing. Fortunately, it's all written out. Fortunately, the important bits are all typed on my screen. All right. I was waxing poetic about the magic of mRNA vaccines, is all that you need to know about that. It codes for specific proteins. All right, and finally, rRNA, or ribosomal RNA. rRNA molecules combine with protein to form ribosomes, and these are part of the translational machinery. Ribosomal RNA molecules combine with protein to form ribosomes, which serve as the base for interactions between messenger RNA codons and tiny RNA anticodons in translation in the cytoplasm. Given your understanding of transcription and translation, fill in the blanks below and indicate the 5' and 3' ends of each nucleotide sequence. Again, assume no RNA processing occurs. So we're going to go directly from a non-template strand of DNA to a template strand based on that non-template strand. We're going to go from template strand to messenger RNA, and then from messenger RNA to anticodons on complementary tRNA. And I think I'm going to write this one by hand because it's a little bit easier for my brain. So going to the template strand, what color are we in? Black, that's fine. We can mark 3' because it corresponds to 5'. A goes to T, T goes to A, G goes to C. That looks a little too much like a G. C, T to A to T, T to A, G to C, C to G, C to G, T, T, A, C, G, T, and 5' corresponds with 3'. Now to messenger RNA, there is an important difference here. There's no, is it thymine? What did I say before? There's no T's. Thymine. There's no thymine in RNA. Instead, we have uracil. So we get 5' again. That's the same. T still goes to A. A goes to U, uracil. C to G, A to U, T to A to U, G to C, 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 A, U, G, C, A, 3'. Finally, we get anticodons on complementary tRNAs. 3' again, always alternating as we go down the steps. RNAs still don't have thymine, so uracil. A, C, A, U, A, C, G, G, U, U, A, C, G, U, onto 5'. Oh. And hopefully if we made any mistakes, there's an enzyme hanging out that will correct it for us. Consider a template strand of DNA. Use the codon table below to answer the following questions. All right. If this DNA strand produces an mRNA, what does the sequence of mRNA read from 5' to 3'? So I'm going to be sneaky and just do it up here. We're going to RNA. So T to A to U, C to G, G to C, C to G, A to U, U, C, G, U, U, A, U, G, G. It's my dog in the background. G to C, A to U, G, C, U, U, 3 prime. That's got to be my housemate coming home. Can I copy this? Pretty please. Cut. Paste. Excellent. I accept. Cool. For what sequence of amino acids does this mRNA code? And assume it does not contain any introns. So, looking at this, I'm just going to go with my highlighter here. Highlight 3, leave 3, highlight 3, leave 3, highlight 3, leave 3, highlight 3. Because I did not do as good a job of leaving spacing as they did up here in the prompt. And so highlighting we can then check, because each codon corresponds to 3 of these bases. And it is worth doing the exercise, but I'm not gonna. And I am just gonna leave these off. So the sequence of amino acids that these codons code for would be methionine, arginine, serine, leucine, tryptophan, leucine, and leucine. You will notice that CUG and CUU both can code first RNA base, second RNA base. Both can code for leucine, which is here. CUU and CUG, I think is what I just said. Yes. What this means is that the codons have redundancy built in, but they're also non-repeating. I want to get that language exactly right. Hang on one second. Redundancy in the genetic code means that most amino acids are specified by more than one mRNA codon. Yes. Yeah. Most amino acids are specified by more than one mRNA codon, but one mRNA codon cannot code for more than one amino acid. So it's specific and redundant. And that is a really important feature of RNA that you should know. I don't know what's happening. Okay. Moving on. The chart lists 5 point mutations, so points being individual points in a strand of DNA that may occur in an original strand of DNA. What happens to the amino acid sequence or protein produced as a result of each mutation? Note that position 1 refers to the first base at the 3' end of the transcribed strand. The last base in the DNA strand at the 5' end is position 21. So let me draw that here. So this, how about not with that? This adenine is position 21, and over here is position 1. There's our original template strand. So if we substitution of T for G at position 8, 1, 2, 3, 4, 5, 6, 7, 8. So instead of G, we had a T. This changes the codon in mRNA to a stop codon, referencing that chart above. Translation stops. A shorter or truncated polypeptide is produced, and this shortened polypeptide is likely to be non-functional. If we added a T between positions 8 and 9, so we're adding it in here. So this is a little bit different. We're not just making a substitution, we're adding an additional position. This will still code for serine. Serine is still incorporated as the third amino acid. But the amino acids that follow are all going to differ because they've been shifted. So this, I think, would be called a frame shift mutation or an insertion. Yeah, this is an insertion mutation. But the amino acids that follow all differ from the sequence in part B above. This is a frame shift mutation. Mutation. I'm going to leave that. Cool. Deletion of C at position 15. So 3, 6, 9, 12, 15. Deletion of C. The first 4 amino acids would be unaffected. But the 5th amino acid would change. The 5th amino acid becomes cysteine, and the subsequent amino acids are also changed in part B. Cool. Substitution of T for C at position 18. Yeah. The original mRNA codon, CUG, that would come from this, and the one resulting from the substitution, CUA, both code for leucine. So no change would occur. And if we deleted the C at position 18, leucine is still inserted as the 6th amino acid in the polypeptide. However, since we're given only part of a sequence, it's uncertain what the next amino acid would be. The peptide, whatever. Sorry about my puppy in the background. I'm not sure if you can, yeah, you can almost definitely hear her. All right, which of the mutations produces the greatest changes in the amino acid sequences of the polypeptide coded for by this 21 base pair gene? Um, we're going to go with the addition of T between 8 and 9. Still leaves the third amino acid intact. However, everything that follows would change. In the substitution of T for G at position 8, a stop codon is inserted and only the first two amino acids are unaltered, and as a result, this mutation produces the... Ah! So, okay, so the addition of the T between 8 and 9 leaves that third amino acid intact and changes everything after. That's a big change. We don't necessarily know what that's going to be, but the stop codon, let's see. Yeah. However, all amino acids after that are different. In the substitution of T for G at position 8, it could be argued that because it's a stop codon, it causes the biggest change. No, because only the first two amino acids are unaltered. Okay, no, I see what they're saying. So basically, we're starting with the runner-up, and then we're going to number 1. The addition of T between positions 8 and 9 still leaves the third amino acid intact, and therefore the one that creates a stop codon in that third position creates the greatest change because only 2 of the polypeptides remain intact. Solid logic problem. I like it. All right, sickle cell disease is caused by a single base substitution in the gene for the beta subunit of hemoglobin. This base substitution changes one of the amino acids in the hemoglobin molecule from glutamic acid to valine. Look up the structures of glutamic acid, GLU, and valine, VAL, in figure 514 in chapter 5 of Camel Biology. What kinds of changes in protein structure might result from the substitution, and explain. So without pulling that up so we can take a look at it, I'm just going to do my best to explain here. So glutamic acid, as the name suggests, is a polar molecule. And as you get into organic chemistry and biochemistry, you'll be able to visualize these a little bit easier. So glutamic acid is very polar, and valine is nonpolar. Being polar, the glutamic acid molecule would have been able to interact with water and other polar molecules. Come on now. There. But the valine molecule cannot. As a result, unlike glutamic acid, valine is more likely to have an interior position in the hemoglobin molecule. Think of your phospholipid bilayers, right? The polar regions, those phosphate heads, face out towards the extracellular fluid and in towards the cytosol. The nonpolar tails, those fatty acid tails, point in towards each other and exclude polar molecules like water. So valine behaves very much the same way because it is a nonpolar molecule. And lipid tails, being made of nonpolar molecules, behave so. All right, why do dentists and physicians cover patients with lead aprons when they take mouth or other x-rays? This should be kind of self-explanatory, I think, but As noted earlier in this and other chapters, X-rays and UV light and many chemicals can damage DNA, and that those replications can persist, or those modifications can persist through replications. So, as noted, X-rays, UV light, and many chemicals can damage DNA. Such damage can result in point mutations and base substitutions, and as we pointed out, deletions and insertions, all of these can have really profound effects in the ability of that DNA to replicate as desired, right? Or as in functionally. And these mutations can cause cancer and other diseases. If they occur in cells that will produce gametes, that is sex cells, mutations can be passed on to offspring. As a result, lead aprons are used to shield the rest of your body from any stray radiation. Lead aprons are used to shield the rest of your body from any stray radiation. So this kind of goes into like the do no harm part of healthcare, and why it's important not to order unnecessary scans if you don't need them, right? We are irradiating our bodies when we get X-rays and other kinds of imaging. As imaging technology improves, right, we can get a higher resolution of detail with less radiation, with less harm done to the cells in our bodies. But the level of harm is never zero, right? So it's a real cost-benefit analysis. Running the test means irradiating this patient's body or injecting chemicals in the form of contrast so that we can see better, or doing repeat scans, multiple angles. All of these things can cause these little bits of mutation, and it's small enough that we decide that it's worth the risk, right? Because the scan may reveal that you already have cancer, or you already have some other condition, a broken bone, pneumothorax, whatever it is. And that is probably a more immediate and severe threat to your health than the relatively small risk of a mutation. And we can, again, reduce harm by controlling how much of you is exposed to that harmful X-ray radiation. So there's my healthcare context spiel. All right. So during DNA replication, which of the following would you expect to be true? Explain your answers. True or false, more ligase will be associated with the lagging strand than with the leading strand. This is true. And this is true because Okazaki fragments are on the lagging strand and would need to be connected by a ligase. Next up, true or false, more primase would be used for the lagging strand than for the leading strand. This is also true. This is also true. Each new Okazaki fragment would be started by an addition of a short RNA sequence by primase. True or false, more helicase would be associated with the lagging strand than with the leading strand. This is false. This is false. Both strands would need to be unwound equally. True or false, DNA ligase links the 39 end of one Okazaki fragment to the 59 end, I think that's supposed to be prime, 3' end of one Okazaki fragment to the 5' end of another Okazaki fragment in the lagging strand. And this is true. This is true. The end of the first Okazaki fragment would be 3' and the next fragment would begin at the prime with 5'. All right. True or false, in the lagging strand, the enzyme DNA polymerase 3, which produces the next Okazaki fragment, also removes the short segment of primer RNA on the previous Okazaki fragment. This is false. A different DNA polymerase removes the primer. All right. Question 12. You obtain a sample of double-stranded DNA and transcribe mRNA from this DNA. You then analyze the base composition of each of the two DNA strands and the one mRNA strand and get the following results. The numbers indicate percentage of each base in the strand. And it just tells us strands 1, 2, and 3. We know one of these is mRNA. Which of these strands must be the mRNA, and explain? Remember earlier when I couldn't remember the name thymine? mRNA cannot produce thymine. It uses, or it is not composed of thymine. It's composed of uracil. So strand 1. Strand 1 must be mRNA because it contains uracil. Which is only found in RNA. Which one is the template strand for the mRNA? Explain. Well, looking at this, strand 2 strand 2 appears to be the complement for strand 1. So our A's and our T's are roughly equal. Our C's and G's are roughly equal. G's and C's. So since 2 is the complement of 1, it must be the template strand for the DNA. Since strand 2 is the complement to strand 1, it must be the template. All right, in a new experiment, you obtain a different sample. Where's my mouse? You obtain a different sample of double-stranded DNA and transcribe mRNA from this DNA. You then analyze the base composition of each of the two DNA strands and the one mRNA strand and get the following results. The numbers indicate percentages of each base in the strand. Which of these strands could be the mRNA, and explain? So they've taken away our primary hint here by getting rid of all of the uracil, which is kind of interesting. So from this, strand 2 has thymine in it, so it couldn't be that. So it could be 1 or 3. Both are complementary to strand 2, and both contain no thymine. So which one must be the template strand for the mRNA, and explain? Since strand 2 has thymine in it, it must be DNA. Let's start there. Strand 2 has thymine. Must be DNA. We can kind of go from there and try to figure out which one is complementary to it. See, which it kind of looks like either one could. It must also be the template strand for the mRNA because it is complementary to both strands 1 and 3. Oh, strand 2 has to be. It has to be the template strand because it is DNA. Yeah, strand 1 or strand 3 could be, either of them could be RNA. The ratios are all similar enough that it's hard to say, but strand 2 is definitely DNA. Cool. That's everything I have for you. Um, Thank you all for your time. I hope you get to rest this holiday. Do study a little bit. Exams are coming up. I'm sure you're going to rock it. Um, Again, not sure about extra credit this week, but submit what you got when you got it. Um, is there a week 10? Yes, next week we will be going over gene expression. So I will see you next week.